Structure and functions of tRNA. tRNA, they transfer amino acid from cytoplasm to the ribosomes where proteins are synthesized. As they are involved in the transferring of amino acid, they are called as transfer RNA or tRNA. Since they are easily soluble, they are also called as soluble RNA. And these tRNA, they are present in the cytoplasm, and each molecule, it is only 73 to 93 nucleotide in length, which means it is much shorter than the mRNA molecule. When transcribed, the tRNA molecules, they are large and they will undergo post-transcriptional modifications. Coming to the structure of tRNA, they show extensive internal base pairing and they will acquire cloverleaf like structure, and they contain significant amount of unusual bases like dihydrouracil, pseudouridine and hypoxanthine. So, these are the unusual bases present in tRNA: dihydrouracil, pseudouridine and hypoxanthine. tRNA, it shows extensive base pairing and it acquires cloverleaf like structure. So, it has an acceptor arm, then opposite to the acceptor arm is the anticodon arm, then it has a pseudouridine arm and DHU arm. In addition to that, it has a short extra arm here. So, acceptor arm is at the 3' end. It carries the amino acid and this area has 7 base pairs. The hydroxyl group at the 3' end is forming an ester bond with the carboxyl end of the amino acid. Opposite to the acceptor arm is the anticodon arm. So, this anticodon arm, it will recognize the triplet nucleotide codon present in the mRNA. So, the specificity of tRNA resides at this anticodon arm, and this has base sequence complementary to the mRNA codon. The pseudouridine arm, it is involved in binding of tRNA to the ribosome. Then the D arm, it is named due to the presence of dihydrouridine in that area. Then this DHU arm or D arm, it serves as the recognition site for enzyme which adds the amino acid. So, the tRNA molecule, it will show specificity in both aspects, in recognizing the mRNA codon as well as in accepting the amino acid coded by that codon. So, this tRNA molecule, they play an important role in translation. They act as adapter molecule between mRNA and the amino acid coded by it. So, what is the function of tRNA? They will transfer amino acid from cytoplasm to the ribosome where proteins are synthesized. So, this is the acceptor arm. What is the function of acceptor arm? They will carry the amino acid. Then opposite to the acceptor arm is the anticodon arm. What is the function of anticodon arm? It recognizes the triplet nucleotide codon which is present in the mRNA. Then you have pseudouridine arm. What is the function of pseudouridine arm? It is involved in binding of tRNA to the ribosome. Opposite to the pseudouridine arm is the DHU arm. What is the function of DHU arm? It serves as the recognition site for the enzyme which adds the amino acid. Next question is, define nucleotide and name two adenosine nucleotide derivative. Nucleotides, they are precursors of nucleic acid, and a nucleotide, it is made up of 3 components: a nitrogenous base, it can be a purine or pyrimidine, pentose sugar, either ribose or deoxyribose, then phosphate group esterified to this sugar. So, when a base combines with a pentose sugar, it is known as nucleoside, and this nucleoside, it is esterified to a phosphate group, then it is called nucleotide. So, a base plus pentose sugar, it is known as nucleoside. Nucleoside plus phosphate group, it is called as nucleotide. Then example for adenosine derivatives include ATP, cyclic AMP, FAD, NAD and S-adenosyl methionine.